There is a region in southwestern Japan where cancer rates are among the lowest ever recorded in modern epidemiological history. Not just lower than the global average, dramatically lower. And for decades, scientists assumed it was something they couldn't touch, something written into the DNA of people born there. That explanation was clean. It was comfortable, and it turned out to be almost entirely wrong. Because when researchers began tracking what happened to those same populations when they moved abroad, adopted different food systems, different sleep patterns, different morning routines, their cancer rates began to climb within a single generation. The genes hadn't changed. The biology was identical. But something in how they were living had shifted, and whatever protection their bodies once carried quietly began to disappear. That discovery didn't just challenge a theory. It opened a door. If the protection was behavioral, that meant it could potentially be learned. It meant the biology behind it could be studied, broken down, and understood. And that is exactly what researchers spent the next several decades doing. What they found is not a miracle cure. It's not a single superfood or a trendy supplement. It's a sequence of morning habits, ordinary, accessible, inexpensive, that appear to influence three of the most critical biological defense systems your body uses to prevent abnormal cells from becoming something dangerous. This is what we're exploring today. Not to alarm you, not to make impossible promises, but because the science behind this is genuinely fascinating, and because the people most likely to benefit from understanding it are the people least likely to ever hear it explained clearly. Your body is not passive. Right now, as you sit and watch this, somewhere between 25 and 50 million cells in your body are in the process of dividing. Each division is an act of biological copying, your DNA being read, replicated, and passed forward. And like any copying process, it carries the possibility of error. Most errors are caught immediately. Your cells have elaborate proofreading systems, surveillance teams, and self-destruct protocols that identify and eliminate damaged copies before they can become a problem. But here is what changes as you get older, and specifically what begins to shift after the age of 50 in ways that are measurable and significant. The enzymes responsible for repairing DNA damage, the molecular machinery that scans for copy errors and corrects them, begin to slow down. Research suggests this repair capacity can decline by somewhere between 20 and 35% compared to earlier decades of life. That might not sound catastrophic until you understand what it means in cumulative terms. Thousands of division events happen every second across your tissues. A reduced repair rate means more small errors are slipping through uncorrected every single day. At the same time, a class of immune cells called natural killer cells, which do exactly what their name suggests, actively patrolling your body's tissues and eliminating cells that display abnormal characteristics, become less aggressive, less responsive. They're still working, but their sensitivity to early warning signals decreases. Cells that might have been caught and eliminated a decade earlier are now occasionally surviving long enough to replicate. And then there is a third factor, one that is less well known but arguably just as consequential. Your body's ability to suppress what's called tumor angiogenesis begins to weaken. Angiogenesis is the process by which new blood vessels form. It's essential in healthy contexts, wound healing, tissue regeneration, but it is also the mechanism by which a cluster of abnormal cells, sitting dormant and harmless somewhere in your tissue, can suddenly gain access to a blood supply. Once that happens, researchers at major cancer institutes have documented that a microscopic cluster can expand to a clinically significant mass in a matter of weeks. What kept it frozen and harmless was the absence of that blood supply. The angiogenesis suppression systems in your body were preventing the recruitment signal from being answered. When those systems weaken, that protection quietly fades. None of this is destiny. These are biological tendencies, not inevitable outcomes. And the reason the Japanese morning ritual matters scientifically is that it appears to directly address all three of these weakening systems simultaneously, not through one extraordinary compound, but through a combination of foods and timing behaviors that each target a different vulnerability. Let's start with timing, because this is the piece that most people miss entirely. Within the first 30 to 45 minutes of waking up, your body produces a natural cortisol surge. This is not the chronic stress cortisol that damages your health over time. This is a purposeful, brief, biologically programmed spike that serves as a morning activation signal for your immune system. Your natural killer cells are primed by this surge. Their activity, their alertness, their willingness to engage with abnormal tissue is at its daily peak during this window. The early morning, specifically the first 1 to 2 hours after waking, is when your immune system's cancer surveillance function is operating at its highest capacity. This means that whatever you consume during that window is landing on a body that is already primed and ready. Your digestive system is active, your immune response is heightened, and the compounds you introduce will be absorbed into a system that is biologically prepared to use them. Introduce the right things and you amplify what's already happening. Introduce the wrong things and you can actively suppress it. The Japanese morning practice that has persisted across generations in the lowest cancer rate regions of the country begins with one specific act: the preparation and consumption of high-quality green tea before anything else is eaten. Not green tea as an afterthought alongside breakfast. Not a quick bag dunked in tap water. A specific preparation of matcha or high-grade loose-leaf tea consumed within 30 minutes of waking, prepared at a temperature that most people in the West have never thought to consider. The reason temperature matters comes down to a group of compounds called catechins, and specifically one called epigallocatechin gallate, better known as EGCG. This compound has become one of the most studied molecules in nutritional cancer research over the past 2 decades, and what has emerged from that research is striking. EGCG appears to interfere with the VEGF signaling pathway, the molecular communication channel that cancer cells use to recruit blood vessels to themselves. In laboratory studies, EGCG has been shown to act as a kind of biological signal jammer, binding to the receptors involved in angiogenesis and reducing the likelihood that a dormant cluster of abnormal cells can successfully call for the blood supply it needs to grow. What makes this finding particularly interesting is the selectivity. Studies from cancer research centers in the United States have observed that EGCG appears to trigger programmed cell death specifically in abnormal cells, while largely leaving healthy cells intact. The mechanism isn't fully understood, but it appears to relate to differences in how cancer cells and normal cells regulate certain internal pathways. The abnormal cell is more vulnerable to the disruption that EGCG creates. But here is where preparation becomes critical. EGCG is highly sensitive to heat. Water above approximately 175 degrees Fahrenheit begins to degrade these compounds, stripping the tea of much of its protective value before it ever reaches your digestive tract. Most people making tea at home are using water that has just boiled or come close to boiling. That water is too hot. The EGCG is being destroyed before you drink it. This is not a minor efficiency concern. Studies measuring catechin concentrations in the bloodstream have found meaningful differences based on water temperature alone. Slightly cooled water preserves the active compounds. Boiling water does not. There is one other factor in this preparation that the research has highlighted and that almost nobody in the West applies: the addition of a small amount of citrus, specifically fresh lemon juice. Vitamin C, when present in the same digestive environment as catechins, creates an acidic protective barrier that dramatically improves how well the compounds survive the journey through your stomach and into your bloodstream. Studies have measured up to 6 times greater catechin absorption in participants who consumed green tea with citrus compared to those who consumed it plain. 6 times. For something that takes 2 seconds and costs essentially nothing, this is one of the most impactful small adjustments in the entire protocol. One more note on preparation that is worth emphasizing because it is counterintuitive: do not add dairy milk to this tea. Even a small amount of milk contains proteins called caseins that bind directly to catechins in your digestive tract, forming compounds that cannot be absorbed. The EGCG effectively passes through you without entering the bloodstream. Plant-based milk alternatives, oat, almond, coconut, do not have this binding problem. But dairy, even in small quantities, significantly reduces the bioavailability of everything you are trying to absorb. Now, the second element of this morning ritual addresses a completely different defense pathway, and it does so through one of the most overlooked organs in human health: your gut. The research emerging from the field of microbiome science over the past decade has fundamentally changed how immunologists understand cancer defense. It turns out that a significant portion of your immune system's ability to identify and respond to abnormal cells is not just in your bloodstream or your lymph nodes. It's in your gut. Specifically, it's in the bacterial ecosystem living there. Among the hundreds of bacterial species that colonize the human digestive tract, researchers at one of Europe's leading cancer centers identified a single species as what they described as the strongest microbial predictor of whether a patient's immune system can successfully target and destroy cancer cells. That species is called Akkermansia muciniphila. It lives in the mucus layer of your intestinal wall, and its presence or absence appears to have profound downstream effects on immune activation and inflammatory regulation. The Japanese fermented food tradition, and specifically a food called natto, made from fermented soybeans, has been shown to dramatically increase Akkermansia populations in the gut. Natto contains specific prebiotic fibers, a unique enzyme called nattokinase, and a dense concentration of vitamin K2 that collectively create an environment where this protective bacteria can establish and maintain dominance in the gut ecosystem. A 50-gram serving of natto, roughly 3 tablespoons, is considered the therapeutically relevant dose in fermentation research. The honest admission here is that natto has a texture and smell that most Western palates find genuinely challenging. The fermentation process gives it a stringy, sticky quality and a powerful aroma that many people describe as intensely off-putting. And if that barrier is real for you, forcing yourself through it every morning is not a sustainable plan. There are alternatives. Unsweetened pomegranate juice has been shown to increase Akkermansia populations through a different mechanism, a class of compounds called urolithins that are structurally active in similar ways to the fermentation byproducts in natto. A clinical trial tracking microbiome composition with precise DNA sequencing of stool samples found that pomegranate extract consumption increased Akkermansia populations by 6 times within 3 weeks. Combining pomegranate juice with another traditionally fermented, unpasteurized food, kimchi, raw miso, kefir, or naturally fermented pickles, provides a reasonable approximation of the gut support function that natto serves in the traditional Japanese morning practice. The critical word is unpasteurized. Pasteurization kills the live bacterial cultures that do the actual microbiome work. A pasteurized probiotic yogurt from a commercial grocery shelf is a fundamentally different product from a traditionally fermented one, and the research consistently distinguishes between them. The third element of this morning ritual is the most culturally embedded and the hardest to translate directly, a specific approach to eating that creates a mild, deliberate daily caloric gap. In the regions of Japan associated with exceptional longevity and low cancer rates, there is a well-documented cultural practice of stopping before you feel completely full, eating to roughly 80% of satiety rather than to fullness. This practice has persisted for centuries, not because anyone understood the cellular biology behind it, but because the communities that practiced it simply lived longer and suffered less disease. The cellular mechanism it activates is now understood. When your body experiences a mild caloric restriction, not starvation, not dramatic dieting, but a modest daily gap between what you consume and what would bring complete satiety, it initiates a biological cleaning process called autophagy. The word translates from Greek as self-eating, which sounds alarming but describes something profoundly beneficial. Your cells, when signaled by mild energy restriction, begin identifying and digesting their own damaged or dysfunctional components. This includes cellular debris, misfolded proteins, and what researchers describe as precancerous cellular material, the kind of damaged accumulated junk that, if left uncleared, raises the risk that a cell's replication machinery will begin producing errors. Research from the National Institute on Aging has documented that even modest caloric restriction is sufficient to activate this cellular housekeeping process. You do not need to fast dramatically or count calories obsessively. The simple act of stopping your breakfast slightly before you reach fullness, combined with a meal built around protein, fiber, and healthy fat, appears to create enough of a metabolic signal to initiate autophagy on a daily basis. Over months and years, that cumulative daily clearing of cellular debris represents a meaningful reduction in the kind of damage that accumulates silently and over time. There is a fourth component to this morning practice that sits slightly outside the dietary framework, but is supported by enough biological evidence to belong in this conversation: morning light exposure. Within the first 2 hours of waking, getting outdoor light into your eyes, even just 10 minutes without sunglasses, sends a direct signal to a group of genes in your cells called clock genes. These genes, including ones labeled BMAL1 and clock in the research literature, do something that most people have never considered. They regulate the timing of cell division. They govern when your DNA repair enzymes are most active. They coordinate the daily rhythm of your immune cell activity, disrupting this circadian gene expression, which is what happens when you spend your mornings in artificial light, staring at screens, never making contact with natural outdoor light, is not a minor lifestyle inconvenience. The World Health Organization has classified circadian disruption as a probable carcinogen. That classification is not based on theory. It's based on accumulated epidemiological data showing that people with chronically disrupted circadian rhythms, shift workers being the most studied population, have consistently elevated cancer rates even after controlling for other lifestyle factors. 10 minutes of outdoor morning light costs nothing. It does not require special equipment or dietary changes. And its effect on the expression of genes that directly govern DNA repair and immune surveillance is documented in peer-reviewed literature. Now, having described what this morning ritual contains, it is equally important to describe what silently undoes it, because the research on this is just as clear, and the behaviors that undermine these defenses are extremely common. A breakfast high in refined carbohydrates or added sugar creates a rapid spike in blood glucose that triggers an insulin response. That insulin response, specifically in the context of the morning immune window we discussed earlier, has been shown to temporarily suppress natural killer cell activity. The immune dampening effect of a high-sugar breakfast can persist for 2 to 3 hours. That is the entire biologically high-value morning window consumed not by protection, but by suppression. A bowl of sweetened cereal, white toast, a pastry, a glass of fruit juice, each of these, consumed in the morning, is doing something measurable to your immune defense capacity in the hours that matter most. The habit of adding dairy to green tea has already been addressed, but it bears repeating in this context because the gap between intention and effect is so wide. Someone who wakes up every morning with the genuine intention of supporting their health, prepares quality green tea, and then pours a splash of milk into it is, biochemically speaking, drinking a cup of warm water with minimal therapeutic value. The catechin-casein binding is essentially complete at dairy concentrations most people would describe as a small amount. If you love tea and you want the protective compounds in it to actually reach your bloodstream, the dairy has to go. Replacing whole food sources with supplements is another pattern worth examining carefully. The appeal is understandable. A capsule is convenient, measurable, and feels clinical. But research comparing isolated EGCG supplements to the equivalent compounds consumed in actual green tea consistently shows lower bioavailability in the supplement form. The reason appears to be that catechins in real tea travel alongside dozens of co-occurring compounds, amino acids, flavonoids, polyphenols, that interact with each other during digestion in ways that enhance absorption. A capsule containing isolated EGCG is a simplified extraction of one piece of a complex biological system. The food matrix, the full ecosystem of compounds present in the original whole food, matters in ways that current supplement manufacturing cannot replicate. A 2023 meta-analysis published in a major cancer prevention journal compiled data from 29 independent studies spanning over 1 million participants and found that regular green tea consumption was associated with statistically significant reductions in the incidence of several cancer types, with the strongest associations appearing in digestive cancers, bladder cancer, and breast cancer. A long-term prospective study following more than 40,000 adults for over a decade found that higher daily green tea consumption was associated with substantially lower risk of advanced prostate cancer in men. These are not small studies conducted on animal models in a single laboratory. These are large, population-level observations from decades of careful tracking. On the DNA repair side, a study tracking a specific biomarker of DNA oxidative damage in adults over 55 found that those who followed a morning protocol including green tea and fermented foods showed a 31% reduction in measurable DNA damage accumulation over 12 weeks compared to those following a standard Western breakfast pattern. 31% is not a rounding error. These are the effect sizes that pharmaceutical research programs spend years and billions of dollars trying to achieve with single-molecule drugs. Now, none of this means that adopting this morning ritual guarantees any particular health outcome. The biology of cancer is complex, involves factors far beyond any single lifestyle intervention, and no responsible reading of this research would frame these habits as a cure or a guarantee. What the research does suggest consistently and across multiple independent lines of evidence is that these morning behaviors measurably shift the biological environment in your body in a direction that is associated with lower cancer risk, better immune function, and reduced cellular damage accumulation. In a field where many interventions produce modest, contested, or inconsistent results, that consistency across mechanisms and study populations is meaningful. One practical note for people who find the morning protocol difficult to sustain in its ideal form: the biology of these habits is cumulative, not all or nothing. Akkermansia populations in your gut need consistent feeding over weeks to establish themselves. The anti-angiogenic effect of EGCG is a daily signal, not a one-time event. This means that consistency at a realistic level, doing this 70% of the time, adjusting what doesn't fit your life, finding workable substitutes for the components that are hard to access, will accumulate real biological benefit over months and years. The people who abandon these habits do so almost universally because they tried to do them perfectly and then quit when life made that impossible. A good enough daily habit that you maintain for years will always outperform a perfect habit that you maintain for 3 weeks. If you store matcha prepared at the right temperature in a sealed container in the refrigerator overnight, it retains approximately 85 to 90% of its catechin content the following morning. Make your tea the night before. The compliance benefit of opening your refrigerator and drinking it immediately without measuring and whisking at 6 a.m. is worth the small reduction in potency. And if you live at a higher latitude where winter mornings are dark, a 10,000 lux light therapy lamp used for 10 minutes in the morning achieves essentially the same circadian calibration effect as outdoor natural light. This is not an optional upgrade for optimizing people. It's a practical tool for maintaining clock gene expression in environments where morning sunlight is seasonally unavailable. There is something worth sitting with as we close this out. The Japanese communities that developed these practices didn't do so because they had access to cancer research or epidemiological data. They developed them over generations through the accumulated observation that people who lived in certain ways seemed to live longer, stay healthier, and suffer less. The science came centuries later. What the science has given us is the ability to understand why those observations were right, to look at the molecular mechanisms and say specifically, here is what the fermented food is doing to the gut. Here is what the tea is doing to the angiogenesis pathway. Here is what the mild caloric gap is doing at the cellular level. That understanding is valuable not because it makes the habits more magical, but because it makes them more legible. It transforms a cultural tradition from something that seems arbitrary into something that makes biological sense. And that legibility matters, because people are more likely to do something consistently when they understand why they're doing it. Your body is doing an enormous amount of protective work every single day without you being aware of it. Immune cells are patrolling. Repair enzymes are checking genetic code. Cellular cleaning systems are running in the background. What these morning habits appear to do, at least according to the most consistent available evidence, is support and amplify that work rather than suppress it. They give your biology what it needs to do what it was already trying to do. That isn't a revolution. It's cooperation. And sometimes cooperation is enough to make a difference that compounds quietly, invisibly, and significantly across the years of a life.